you have a connection to Apple?
You don't have, so can you log in Zoom and share your screen via Zoom? Because then it's easier for them to record.
Okay. And the Zoom
ID is this one.
which one?
I can click join. And then 982
982
2363
2363
7053.
53.
Recording in progress.
Okay.
So now hold on. They need to make you a panelist. We got time. So this is in case you're staying for lunch.
Oh, yeah, I'm triple S is, yes, thank you. Okay.
Recording in progress.
Yeah, I joined, I'm a panelist.
Okay, so now you can share if you go into Zoom.
Yeah, I just, four. There's four. What's that? ManiFest. What's that? Dismiss. All.
So Mani will
Go
be
into Zoom. Share screen.
your desktop. Yes, that's great.
Okay. It really is.
If you want to minimize this, you can click here, then it's not so big. Yeah, you can click on this one.
And this. Okay, and there's no temporary mode, I guess. Speaker mode, presenter mode.
I assume I'm not so sure. Sorry, what's the?
Is there a presenter mode?
Oh,
And then
you
I have to,
mute your.
yeah. Is there a presenter mode?
I don't. Sorry, I don't know.
Didn't matter. I'll fake it.
No, it's down in, I think you click on that, no?
Yes.
And it doesn't work?
Now let me see.
There you go.
No, that's not.
Oh, that one, I see what you want. No,
Yeah.
you can't- yeah, but you
Yeah,
can't
don't
do
worry about it.
presenter.
Oh, sorry. Can we hook you up on the mic?
Yeah. So it's really a great pleasure to be introducing Steve Chu. And he's one of those people that I really enjoy introducing because I don't have to bother with all the awards, distinguished chairs, honorary degrees, because there is one prize that stands out above all others. It's, of course, the Nobel Prize in Physics for laser cooling. And he has other noteworthy achievements. He was director of the Lawrence Berkeley Laboratory, served as the president of the American Association for the Advancement of Science, and he served as Secretary of Energy under President Obama. And remarkably, the United States Senate confirmed him unanimously. We, of course, absolutely delighted that he has come here and he will tell us about his recent work. His lecture today will be on molecular motors and non-thermal equilibrium statistical physics. Please.
Okay, thank you. All right. Thank you. Pleasure to be here and celebrate Manny's 90th birthday. Yeah, that was an unusual confirmation. It was unanimous, and it was also the first confirmation of the first scientist cabinet member. So And neither will happen again. Okay, so I realize I'm talking a bunch of mostly theoretical physicists, and I want to talk about biology, so I'm going to ease you into it a little bit. So I'm going to play a few little vignettes of what's happening, and let's see if this works.
Constant motion inside of your cell. There's new construction, demolition, and most importantly, transporting goods from one place in the cell to another. Cells transport goods along cellular roadways.
These are the molecular motors that do all these things.
The cells use motor proteins. Kinesin is one of these motor proteins. If you didn't have kinesin and other motor proteins, you simply wouldn't be alive. All the cells in your body depend upon these tiny motor proteins to organize and power themselves, to divide and multiply, and to communicate with other cells. Let's start with how you began.
Well, we'll start to end there. So that's an introduction to the motor proteins. So we're going to be studying motor proteins, and I'm going to show you a well-known cartoon of how a motor protein is supposed to work. It does not work this way. This is kinesin carrying this vessel, this big blue thing, along a microtubule, that cylindrical track. And it looks like a macroscopic machine that's well programmed. But in truth, this is a bit of fiction. Yes, it has steps that go over and over, but it does not look that way. It does not operate that way. All right, so I'm going to begin by quoting the greatest American philosopher of the 20th century. In case you're wondering, it's Yogi Berra, seen here in a philosophical conversation. And he said, you can observe a lot by just watching. And so to that end, I want to show how we developed some new probes. These are rare earth nanoparticle probes. We didn't invent them. They were developed maybe 6 to 8 years before we came along. But in any sense, the red fluorescence, relative fluorescence is shown. They can last for many, many hours at the high luminous intensities without blinking or photobleaching or anything else. And we compare that in gray to the quantum dots, to the best organic dyes, the purple one, or to fluorescent proteins. So they have a lot of advantages. What did we do? Let's see, why does this not advance? That's interesting. So I have to push down here. Okay, there's a little arrow. Okay, and so we published a paper on this in 2018, where we began by collaborating with some material scientists, but they were disappointingly not that bright and all clustered. So in desperation, we learned how to make them ourselves, and they're about 100, 200 times brighter. The system we're going to study, you learned about cells and how cargoes are transported within cells. These are essential to the survival of a cell. And two of the main motor proteins are kinesin. That's the thing that you saw walking hand over hand on that microtubule. And there's another major protein called dynein. And in dynein, the cargo is transported the other way. So if this is a neuron in the cell body, kinesin will take cargoes out to the distant axon ends, and that dynein will take the cargoes back to the cell body. Kinesin, Ron Vale, who was narrating that film, discovered kinesin in the 1960s, and by now it's fairly well understood how kinesin works. Dynein was discovered much earlier, and yet because it's much, much larger, much more complex, the details of how it works are not understood yet. This blue thing, these two legs, and all these blue attachments are one dynein molecule. It's 1.5 million megadaltons in mass, and they have lots of accessory proteins. So what do? We put in a little flow cell, microfluidic cell, and we take immature neurons, and we induce the neurons to grow the axons along a long observation chamber. And finally, they come out on the other side. And what we're going to show is data of 12 different vesicles tracked over 900 microns, taking 100 video frames per second. And the journey takes roughly 20 minutes. So we're looking in live neuron cells. This is another picture of it, and this is an example of some early raw data where you see the emission from these nanoparticles. There is no background because we're exciting at 980 nanometer light. It's in the infrared. There's no cell autofluorescence. Sometimes the motor gets a little confused, goes backwards, it goes along. Sometimes it gets even more confused, goes onto another different track, and then really goes backward. So this is the raw data, and you're about to see it go off on another track. And so we can track this to about a few nanometer precision, as the noise is about 4 nanometers. Okay. One of the reasons why we undertook this study is because one of my postdocs was interested in Alzheimer's disease, and there was a supposition perhaps that people with different forms of protein called ApoE, ApoE2, 3, and 4, and if you have ApoE4 in your genes, you have a 4, 5, 6 times higher chance of getting Alzheimer's in an earlier time. And so the question is, does transport of dynein and kinesin, is it affected if you have these different so-called alleles? And the wonderful thing about biology is you can take, let's say, a fibroblast from a human patient who has these, or other Alzheimer's mutations, and grow neurons, human neurons. So we do our experiments on rat neurons and also on human neurons. Okay, the first thing we're going to do is very simple. Here you see a plot of time versus distance. And as the motor moves along, sometimes it pauses. We don't know why. And it begins to launch on a trajectory at a certain roughly constant velocity. Perhaps it goes to another constant velocity. And what we're going to do is we're going to look at the distance it goes in a certain time tau from xt to xt plus tau, starting at this position and along all straight trajectories. And this is one example at time t equals 0. The motor is here after time 10 milliseconds. There's a distribution of where the cargo could be. Sometimes it goes backwards a little bit. And so that's the negative x's over here. But most of the time it goes forward. After 40 milliseconds, 100 milliseconds, it looks like roughly a Gaussian distribution marching in the right direction. All right, this is a distribution. It has a mean, it has a standard deviation. And so we're going to define a quantity phi as twice the mean divided by the standard deviation. Okay, S squared is our standard deviation. And what we find surprisingly is in this one particular vesicle, it moves along and reaches some asymptotic plateau, continues there for a while. Another trajectory, same vesicle, it moves along, finds another plateau, finds another plateau. It's erratic in which plateau it finds. And it doesn't do this all the time. Sometimes it goes on an erratic thing and never plateaus. But the majority of cases is forming this plateau. Well, that's one vesicle. And so if you look at 12 vesicles, which are all different, you find that some vesicles, the one I just showed you, had 3 plateaus. Other vesicles only had 2 plateaus. Some had 5 plateaus, and so on and so forth. So each vesicle had a different number of plateaus. But if we took all this data with the different size vesicles, the very different behavior, and just simply formed a histogram of where these plateaus are, the quantity, remember, is twice the displacement over sigma squared. So it has the dimensions of 1 over distance. And in this case, inverse nanometers. You find a beautiful quantization of plateaus. No matter what the vesicle is, the vesicles are all different, all 12 vesicles, and yet there's this quantization. So the question is, what does this quantization mean? Okay. Well, there's a hint in electron microscopy from many decades ago. Here's the vesicle, here's the microtubule. And at that time, they didn't know what these things were. The authors called them cross bridges, that bridge between the vesicle and the microtubule. And in this, the dark bars are the dyneins, which are taking the material and going back to the cell nucleus. So that's called retrograde transport. And they see some of them have 0 connections, but 1, 2, 3, 4, 5 connections. So if you look at the 1, 2, 3, 4, 5 connections, they're strikingly similar to the number of plateaus. So one possible explanation is these plateaus actually govern how many motors are being used by the vesicle to move the cargo backward, back to the cell nucleus. So that's the conjecture. But can we do some more than just say maybe this is so? And the answer is yes. Let's suppose that each motor system is attached to a different part of the vesicle and acts independently. And let's also suppose that a motor system, after a certain amount of time, certain displacement x, it actually has, could displace this vesicle by some probability distribution that looks roughly like a Gaussian. So P1 of x is this Gaussian distribution after the mean has moved x. If they're independent, the product motion will be just the product of the two distributions. P is equal to P1 times P2. And if these are Gaussian distributions, you can show analytically that the product is a Gaussian. The mean is not any faster, it's the same mean, but the dispersion goes down by a factor of 2. Now, that seems counterintuitive, but once you understand it, it seems really obvious, which is always, if you know the answer, things seem obvious. It's a golden rule in physics. So here's the vesicle, and there's a motor, and it goes forward, but this other motor hasn't moved. And so what does the vesicle do? It splits the difference. So this one moves forward, this one hasn't. It splits the difference. So the motion is half as much, but it's occurring twice as fast. But because the steps are half the size of the steps, it gets smoothed out. And so that's what the math is saying. And for n independent motors, it's just the dispersion goes as 1 over n. And so if you look back at this thing, you have this beautiful quantization. We think that this is really the number of motors that the system is using. Why biology chooses to change between 1, 2, 3, back to 1, 2, wobble around, we don't know, but it does it. All right. There's another prediction, and that is the mean doesn't change. So we look at the average, there's a big, huge velocity dispersion. If this is 1 motor, 2 motors, 3 motors, the speed of this one motor can vary all over the lot, 6 microns per second down to 1/3 of a micron per second. We also don't know why that is, but it's similar with 2, 3, 4. This behavior was also seen in experiments where you take this motor system and put it in vitro system where it's artificially constituted on the surface of the glass. You know the motor's there. You try to assemble all the parts, and you do similar sorts of things. So in our data, there's only a very, very weak dependence from the number of motors. But in an in vitro experiment, you can know precisely how many motors you put on. So a single motor attached to a piece of DNA. Why DNA? Because you use the DNA to bridge 2 motors, 3 motors, 4 motors. And what you find for 1 motors, 2 motors, there's no difference. By the time you get to 4 and 7 motors, they begin to get in the way of each other. But this confirms that, and it doesn't go any faster. So it's going the same speed, but we're finding the distribution decreases. Okay. Now, there's something else about these probes that are good. They're very, very bright. They are emitting roughly 100 times more photons per second than a quantum dot or an organic dye, 100 times faster, which means you have 100 times better resolution. And for that reason, when you're in a live cell, you can actually see individual molecular steps. So these black lines are a computer program that's trying to decide, did the motor take a step? And if it did take a step, how far did it step? And so taking it at room temperature, 22 degrees up to 37 degrees, the temperature of the cells, we find these funny distributions. So for the moment, ignore the black lines and just look at the data, the histograms. And this point over here is 0 steps. The motor hasn't moved. We don't measure 0. So this is roughly centered at 8 nanometers, but it's really not fully resolved at all. And this is 8, 16, 24 nanometers, -8, -16 nanometers. And as you raise the temperature of the cell to body temperature, you find that the backward steps almost disappear, and it's mostly taking steps around 8-ish nanometers. Okay, so it's taking roughly 8 nanometers. Okay, so what's going on? Well, I just told you we have 1 motors, 2 motors, 3 motors. So how do you know what we're looking at when we're looking at single steps? There you go back to the beautiful in vitro data. And this is our distribution at 22 degrees C. This is the distribution of an in vitro experiment at room temperature. They didn't specify, but roughly 22 degrees C. They look very, very similar. So that's good news in 2 respects. We know we're looking at dining. We know we're looking at single molecule. And the guys in vitro systems got most of the parts right. So it seems to be marching along with the same distribution. So that gives us confidence that these single steps are of 1 motor only, which makes life easy. So this is a cartoon of, again, by Ron Vale. Okay, so this is not as goofy as the first motor. It's showing that the 2 legs of the motor are diffusing a bit. When the thing is jiggling around, it's diffusing, and this little foot down here, over here, it's landing on microtubule. It's docked on there. And so this is a 1 dining monomer. This is a long step. This is a very long length. This is 15 nanometers. And this thing over here, these orange things, connect back to additional molecules that finally connect to the vesicle. And you saw these 2 motors. What happens is, see if I can do this without falling over. 1 motor, another motor. Lifts up, it kind of goes and jiggles around, and then finally starts to land. And then the linker arm changes, and it just took a step. The next motor jiggles around, it can step, or it can step backwards. And so you have these 2 legs jiggling around. And the question is, that's a nice heuristic argument of how this motor works, but can one quantify it? So we have some data that will help quantify it. And so again, this is displacements, distributions in displacements, 22, 30, 37 degrees. The black curve is a fit to a Gaussian where we're saying, well, we don't see 0 steps, but we know they're occurring. So we'll do the Gaussian fit and center the Gaussian so it encompasses the 0 thing, which we don't see. And so if you notice this, the first thing you notice is when you go from room temperature to 37 degrees, the width of the Gaussian shrinks. The other thing that's very surprising is the center of the Gaussian does not change. It remains at 5 nanometers. And so how do you interpret that? Well, you do exactly, you make a mechanical model. You say when it's up, it gets to diffuse. Okay, you use our measurements to say what's the range of diffusion, but then it puts its foot down, but it doesn't really take a step. It can take up and jiggle around a little, take a foot down, goes up, and it does this until some chemical event happens that makes it irreversible when the foot is down and then it launches forward. So if you look at that and it's doing all this, then it moves, and then we measure the displacement. And you begin to ask, what's the physics of this thing jiggling around? Well, it's an object in a viscous fluid. You look up what the viscosity of fluid is. You leave as a free parameter what the effective spring constant is. The range of the spring constant is the range of motion we see, we measure, and the rest is temperature and everything else. And so this is the solution to that overdamped diffusion of this leg moving around trying to take a step. The amazing thing about this is you plug in all the numbers and you look up the viscosity of the cell and everything, and to within 30%, it matches. And these single set of parameters, the length of the spring constant, namely the parameter we're determining, lie, and these are different temperatures. And this is the time the dynein spends in an unbound state. As you look, if it's a longer time, what happens is the following. This thing goes around, it goes up, goes up, goes up. Okay, nothing happens. And there's a chemical event that thermally desorbs. So at low temperature, that chemical event is slower. So we just leave time as a free parameter, and what you find is a simple model fits all three data very, very nicely, and it fits the known size of the molecule and the known viscosity. So I said, okay, this very, very simple elastic spring model seems to work. What more can we say about this? Well, go back to the single steps. Now, in these panels D, you see a rise and a fall. What are we plotting? We're plotting, it made a step, and then there's a delay time before it makes another step. So these histograms are the time between steps, again, determined by a computer program. And when you go to higher and higher temperatures, the character of the curve does not change. And in fact, the black line is a fit to a distribution, tau times some rate constant, k squared e to the minus tau dt. Where did this come from? If there are two events in this motor, and one of them is described by just a simple rate constant, but then it's followed by another rate constant, then the dwell time is just a sequential two rate constants with some, k, and that's a convolution, and that's how you get this. And it turns out these rate constants are equal. They're not, okay? So now we have rate constants. It's two rate constants, not one. All right, here comes the first thing that you know there's going to be trouble, because it's now going to be in conflict with the accepted model of dynein over the last 2 decades. In each site on this molecule, there's sites where ATP can bind and it can burn. So in biology, ATP is one of the units of fuel, the major unit of fuel. APO means the site is empty, ATP binds. Then after it binds, the molecule closes around it. There's very rapid electron transfer. And typically what happens is ATP hydrolyzes by breaking a bond, a phosphate bond of the 3 phosphate groups on ATP. So it's 2 phosphates, ADP plus a phosphate, but they're still stuck together. This phosphate then leaves. Once this phosphate leaves, then allows ADP to leave, and now it's in the empty state and it cycles all over. Okay, that's one ATP cycle. But we have K1 and K2, so we have 2 ATP cycles. But let's start with what this could be. Of this ATP cycle, these are the only possibilities. Well, at a cell concentration, the concentration of ATP is about 3 millimolar. And we know what the binding rates are from earlier experiments. It takes about a millisecond to bind. In the in vitro experiments, because their probes weren't as good, they took data around 10 micromolar instead of 3 millimolar. So they slowed things up so the time between steps is really long. And so there, the rate limiting step is ATP binding. In our case, it's not the rate limiting step. Hydrolysis, we know, occurs really fast, less than a millisecond. We also know from previous kinetic experiments that ATP, when it leaves the site, it leaves in less than a millisecond. So by default, there's only one process that's allowed that has this time scale. And it's the phosphate group that has been cleaved off. How long does it take to leave the molecule? Okay, that's great, but there are 2 of them. That means there have to be 2 phosphate groups. And now this is beautiful because thermal desorption is thermally activated. And so yes, does it fit thermal activation? The answer is yes, beautifully. And Arrhenius plot says, our 3 temperatures say it's something being thermally activated. We're the first experiments to actually change the temperature in vitro or in vivo. It wouldn't have mattered with the in vitro guys because the rate limiting step was the on rate of ATP, which is very temperature insensitive. Anyway, so we now know it's 2 constants. Now, I'm not going to go into the details, but this is a crystal structure of one of the dyneins. This is 3 AAAs. There's these little segments here are so-called AA1, AA2, AA3. There's a long stalk, and this little knob up here binds to the microtubule. And in the previous model, when ATP lands at this single site between AA1 and AA2, these 2 domains of the molecule close by about a nanometer. And the mystery is, can you understand how this closure of 1 nanometer when ATP binds enables this thing to take a 10 or 15 nanometer step? There's many things, but I'm not going to go into that because I want to go into some statistical mechanics. But what we're finding is we need 2 ATPs per cycle, and the only 2 real sites have to be an AA1 and AA3. Okay, so over the last 3 years, a postdoc and me didn't spend any time taking the data. We spent the time reading the literature, all the supplement materials we could find of everything, and read the supplement materials backwards, forwards, and everything else, and developed a model using 2 ATPs. And it seems to be more consistent with our data and our model than the earlier model, and more consistent with the biochemical measurements. But, and there are reasons why the analysis of some of the other data was a little off that we have pointed out. But the most important thing about this model is it tells the structural biologists you can do these experiments and see for real, is it one thing or the other, and also in vitro data, and also further on carry on experiments. That's the model. I'm not going to talk about it. The model talks about small movements of little peptide chains that weren't noticed. And the beautiful thing about biology is the structures are in something called the protein data bank. And so we were able to take the structures in the protein data bank, look at what's happening. While most of the biologists were concerned at the site AA1, AA2, we were also looking at AA3, and the data they took actually confirms our model, but they're more definitive experiments. So that's the real cool thing about biology. It's like astronomers. Once the data is out there, anyone can look at it. Okay, let's move on. I want to talk about a question Erwin Schrödinger posed in a series of lectures he gave in Dublin in 1944. He says, is life based on the laws of physics? Of course, you say, of course, everything's based on the laws of physics. But he said, from all that we've learned about the structure of living matter, we must be prepared to find it working in a manner that cannot be reduced to the ordinary laws of physics, not because there's any new force, but because the construction is different from anything we have yet tested in the physical laboratory. All right, so let's think about that. Suppose Newton had Newton's brain, but was the size of a bacteria. His laws would be very different. An object in motion would completely come to a rest in sub-nanoseconds if it's a micron scale particle. But it doesn't really come to rest. It sits there and just kind of jiggles around. There's a hand of God just seemingly jiggling it, Brownian motion. F does not equal ma. There is no inertia at this scale, but the forces are dependent on, for example, Stokes drag on the surface area. So very, very different forces involved in nanoscopic dimensions in fluids. And these motors not only include the motors I'm talking about, they include all the things that make life work, like transcribing DNA into RNA, translating RNA into amino acid chains that fold into proteins, everything. Okay, so now let's now think about this motor as a motor and step back and say, what's the efficiency of the motor? Well, we know now it's one motor. We know how fast it can go. We look at the fastest motor. We also say it's hydrolyzing 2 ATPs, not 1, but 2. And we look up the concentration of ATP and ADP in a cell, and we realize for an ADP, it's releasing 5 × 10⁻20 joules per ATP, okay? And so that's great. So for every time the enzyme walks in a full cycle, 2 ATPs are burned. We know the viscosity of the stuff in the cell. We know how fast it's going. So we're saying the chemical energy does 2 things. It overcomes the internal friction of the motor and makes the motor click. But in the end, it's actually doing mechanical work by pulling this big bag through a viscous fluid. And if you say, what's the energy of pulling this big bag through the viscous fluid? The motor is 45% efficient, lower limit. It could be higher. Okay, that's pretty good because it's working at 1 temperature. It's not a heat engine. Okay, now, so then the next question is, how's it so good? And what's its real limit? Its real limit comes later in a future experiment, but we have a lower limit on how good the motor is. Okay, and the difference in this motor versus that cartoon of Kinesin is that that cartoon of kinesin does looks very developed, very, you know, it looks like a macroscopic motor just munching through like this, where in fact it's just jiggling around and doing all sorts of things. And so in a living organism, the molecular machinery is embedded in a viscous fluid, friction and thermal fluctuations dominate everything. Oops. And again, I won't go into it, but a detailed version of how clever this motor is has emerged, and we haven't written it up and submitted it yet, but it's very similar to my studies done 15 years ago on the ribosome, translating messenger RNA into amino acid chains, and how the ribosome uses statistical fluctuations to get very good accuracy in the translation. And this is how it actually can use little binding events and little pieces of energy. The limit, by the way, the theoretical limit, I'll discuss in a moment, is about 95%. It comes from a very different thing. Okay, let's talk about statistical physics of the Dynamo motor. First, I want to tell you about the fluctuation theorem. And consider two molecules colliding. Time is going upward in this case. They collide and they bounce off. You reverse time and they collide and they go the other way. The equations of motion are time reversal invariant. It looks the same going backwards and forward. Very, very different if an egg drops on the floor and splatters. We teach our students this in the first statistical mechanics class and say, this is just the laws of large numbers. This is a very simple thing. You have many things. There are many ways of distributing where the egg is once it's crashed on the floor. And so along comes a theorem called the fluctuation theorem that quantifies this, and it gives the probability of entropy flowing increasing, as in the splattering egg, divided by the movie going backward, so entropy flowing backward, is equal to E to the change. Sigma is the entropy flow, T is the time lapse. So sigma T is the change in entropy. Divided by K, K is Boltzmann's constant because temperature is not measured in joules, it's measured in degrees. So anyway, it just says that the probability of going forward, movie running forward versus movie running backwards, is exponentially large as the time increases. So it quantifies this notion. This theorem was discovered in 1993 through a numerical simulation and proven a year later. So not everything in StatMech was done in the 1800s. And this theorem actually doesn't violate the second law. It says you're allowed to go backward in time for brief moments because it's just laws of large numbers quantified, all right? But the surprising thing is this theorem operates for small micron-sized things where there are still billions and trillions of events going on, okay? And the most remarkable thing about the theorem is it doesn't require a thermal equilibrium. It's just plain true, whether you're in and out of thermal equilibrium. Okay. All right, so now let's take the fluctuation theorem. This is movie running backwards versus forwards. Going forwards is normal displacement, backwards is going backward, is equal to this. And we consider two paths, but this is a probability distribution of paths being taken. So let's take two out of an ensemble, and you go that way. You consider the time reverse one, so I've offset them a little, that's time reverse. But if you start everything at t equals 0, you project them to here. So you get two paths going upward in time, forward in time, or two paths going backward in time. So the backward in time movie, we are constructing algebraically, okay? Because we're burning ATP, that's a lot of degrees of freedom, and you'll never see anything go backward when you burn something. But so we can construct it algebraically, and we find that the log approximates the Gaussian distribution, as I've showed you, and you can construct the backward time. And the great thing about the log is when I take the logarithm of both sides, I can get, this. The logarithm of the probabilities is now just a change in entropy over K. And this is our old friend, 2 times the displacement divided by sigma squared. So that's why the 2 is there, just in case you were wondering. It was motivated by the so-called fluctuation theorem, all right? But you don't need the fluctuation theorem to say what's the displacement divided by the dispersion squared. But you do need the fluctuation theorem to relate it to some thermodynamic properties. In particular, it's a statement about the change in entropy. So if it's a change in entropy, and suppose suppose temperature is a good parameter, i.e. you're in thermal equilibrium, then you can say this is effectively the amount of heat entropy you need to do something. So then you go back to elementary stat mech and say, okay, dq is equal to the enthalpy change plus the work done. But the enthalpy change of a full motor going through a full cycle burning ATPs is it always returns back to the same spot. So therefore, delta H is 0. Delta work is easy, that's force times distance. You stick in all the numbers, and you find that our measured quantity phi is equal to the average force, drag force, the Stokes drag force, divided by kT. Now you do some arithmetic, and if the cell is at body temperature, 310 Kelvin, 37 degrees centigrade, you find that this force, the average force looks like 0.43 piconewtons. That is in direct violation from what we know of the viscosity of the axon and the size of the cargo. Because from that, we're saying the average force given the velocity should have been 2.45 piconewtons, not 0.43 piconewtons. Okay, what's going on? Everything was great until now. And what's going on is you look back at this thing, and you see there's the force, Stokes drag, measured quantity phi. We can look up what Boltzmann's constant is. That hasn't changed much. And what about t? If we let t be a free-flowing parameter, and t is 0.57 times 310, then it would be equal. So what does it mean to have t being 5.7 times cell temperature? You know, things at that temperature, that's pretty hot. Things get ionized. Okay. Now I'm going to remind you of another theorem called the Fluctuation Dissipation Theorem, which relates Brownian motion to Stokes drag. Einstein kind of finessed it, and this is his Brownian motion and mobility, but it was first proposed by some people at Bell Laboratories, of course, as a general description between fluctuation like voltage fluctuations and Johnson noise in electron conductivity in resistors. It shows up in many areas of physics. Okay, so the fluctuation dissipation theorem is a mainstay in equilibrium thermodynamics. And in 1931, Lars Arnsager, a chemist, physical chemist, said, suppose there's a fluctuation and suppose you put the system out of equilibrium, a small perturbation out of equilibrium, and watch the way it relaxes to equilibrium. He says the way it relaxes to equilibrium is the same as the fluctuations you see in thermal equilibrium. And that was called the Arnsager regression hypothesis. Now, in the 1960s, he gets a Nobel Prize for this work because it's the first brave venture out of equilibrium thermodynamics to see what do systems do in non-thermal equilibrium. In hindsight, maybe not so mysterious, because if you consider a thing in equilibrium, but suppose a fluctuation takes it 3 sigma away, you start the clock there, and you can't say whether I took the system out of equilibrium, put it here, let it go, and relax, or it just relaxed back to the norm. So in any case, it's a theorem, and it was worthy of a Nobel Prize. So now the fluctuation theorem, I'm going to skip the math, but the fluctuation dissipation theorem generalized has a very simple interpretation for a very special case. And what the special case is called steady state non-thermal equilibrium. And the essential ingredient of steady state non-thermal equilibrium is you can't tell time going forward and backward. In thermodynamics, there is no time. It's just statistical distributions, and you're taking averages of distributions. In non-equilibrium, of course, time is going in a particular direction. It's being driven by something like, you know, chemical energy. And in this region of steady state non-thermal equilibrium, where time invariance comes into play again, one can show that 1 over the effective temperature is equal to delta C is a correlation. If a particle is here at a certain time, it's here at a certain time, and you make a statistical average of the correlation, that's C. And this chi is, well, if the particle got a couple of steps, how does the system integrate those couple of impulses to have a displacement? That's the formal thing of what happens. And when this thing reaches a constant slope, it becomes a constant temperature, okay? But let me just say that's a lot of math. The fundamental thing is time reversal invariance. If you reach a steady state, you can reach a steady state and you get an effective temperature. This is a stupid, I'm an experimentalist, by the way, I'm not a theorist. Thank you for having me anyway. But this is my version of what this all means. In statistical mechanics, one of the most powerful things is you have an equal partition theorem, which says for every degree of freedom, let's say a gas molecule in the air going and say along the x dimension, the mv squared of this kinetic energy along this direction is equal to 1/2 kT. You get a y squared, you get a z squared, you get rotations, you get all sorts of things. If it's a thing on a spring, there's another degree of freedom. Okay, each degree of freedom gives you 1/2 kT. Now let's look at the basic physics of this motor. So it's the Langevin equation, which loosely speaking means F equals ma. What's the force? The ma is the mass times the change in velocity. What are the forces? Well, there's the force of the motor. Once the motor gets something to move, there's a viscous drag force. And then there's the invisible hand of God making the thing jiggle with Brownian motion. So now I divide the motor force into two parts, an average constant force and a jiggly force, because this is a stochastic. When the motor decides to make a step is determined by thermal desorption rates and other things. So the motor now has a steady state, average force and a jiggly force, drag force in this. Why do I want to do that? Well, if I go to the center of mass frame of the motor, all of a sudden the motor is making the vesicle go at a certain speed. I go to this center mass frame, and in that center mass frame, the drag force and the motor force go away. So now what I have in the Langevin equation is something very simple. It looks like mv, change in mv is equal to the difference around this mean moving force, and that's the drag, and two jiggly terms, the normal fluid jiggly term and the motor jiggly term. Because in this moving frame of reference, instead of stepping with some stochastic thing, it looks like it's stepping forward half a step or stepping backward half a step in some stochastic way. So it looks like it's jiggly. And in this moving frame of reference, after many, many jiggles, what happens? It goes more jiggly, 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 and the distribution widens. So it looks like it's diffusing. So in the frame of reference, you begin to see diffusion. Now, when does the motor overpower the fluctuations of fluid? Easy. Big vesicle, 100 nanometers, small jiggles. Mother motor, when I say step, you step. When I say you step. So the motor can simply overpower the fluctuations of the fluid. Therefore, the fluctuations can easily become bigger than the fluid fluctuations. What's the condition? Well, we know from elementary statistical mechanics that the correlation function between the fluid jiggling at 2 times T1 and T2 has to kind of appear like a delta function. In real life, it's not a delta function. It has a kind of a correlation about 10 minus 14 seconds because there's a lot of atoms. In this motor is not stepping once every 10 to 14 times a second. It's stepping every 20 milliseconds. But here's the trick. If you average over 50 milliseconds, 3 steps, and take 3-step blocks, it becomes time reversal invariant. It looks like noise. So the effective temperature in this case, you'll have to average over a certain time, and then it has the delta function appearance all over again. And so when you do that and do the math, sure enough, you get the right answer. Anyway, that's the elementary way. If you want to see the way theorists do it, you're going to have to spend days and days and days reading about it, but this is actually a better way to think about it. And not only that, it actually tells you what the necessary and sufficient conditions of getting out of thermal equilibrium are. The motor's got to do it at a certain rate, and it's got to be powerful enough to overcome the viscous drag. And once you achieve that, you're out of thermal equilibrium. All right, let's continue. Turns out this has been studied in an analog way with an artificial system. People took an optical tweezer, on a bead, and they just jiggled it. And they jiggled at different rates and in different amplitudes. And they said, sure enough, all the stuff of the fluctuation theorem and the fluctuation dissipation theorem out of thermal equilibrium is true. But this is an artificial system. It's just a bead being jiggled by an optical tweezer. It's a great paper, by the way. And we looked at this paper. I read it 5 times and didn't fully understand it. Started reading the outsider stuff and everything, and then developed what I just told you. But in our system, this is the first system that occurs in nature, not an artificial system, where you're way out of thermal equilibrium. All right, there's more. This fluctuation theorem has a corollary. I put down the fluctuation theorem again. The probability distribution of time going forward and time over backward is exponentially large going forward in time. Now, if you take one step and the probability of taking a step is this rate curve piece of 1, and then the probability of taking another step is piece of 2, the distribution probability after 2 steps is the convolution of those 2 functions. And after many, many steps, it's n convolutions. For any integral function, n convolutions gives you a Gaussian-like function, at least in the central part. That's even more powerful than the central limit theorem, but it's Gaussian-like. Okay, now let's define normalized dispersion. Dispersion is sigma squared, and displacement is just displacement. So we normalize it, so it's a sigma squared over twice the displacement times the number of steps. And it's possible to show thermodynamically that delta Q, that's T delta S, times this normalized dispersion is equal to 2 kT if the distribution is Gaussian. And in general, if the distribution is not Gaussian, it's greater than 2 kT. So let's just pause here. The fluctuation theorem is derived in an uncertainty principle, which says the product of the amount of heat you need to do something and how orderly you can do this thing is a minimum product of 2 kT. Let's put it on something you know about. Suppose you have a quartz watch. Quartz watch has an accuracy of about 1/2 second a day. And you ask, what's the minimum amount of heat energy you need to run this watch for a day in order to achieve this accuracy? And it turns out it's, well, accuracy is different than precision. Accuracy is absolute. Precision is 2 orders of magnitude more precise. And if you do that, it requires for this Mickey Mouse watch, this was actually a paper that analyzed a Mickey Mouse watch, 5.8 times 10 minus joules to keep this watch accurate over 1 day. For the most stable atomic clocks, there's 22 digits. So you need a lot more energy. Okay, so that's where the lower limit is. It doesn't say what the watch is. So when I said the motor can be 95% efficient, this is the entropy needed in order to do something at a certain orderliness. It gives you this 95%. It's a thermodynamic limit. It's very similar to the limit that Landauer suggested and sort of derived, that if you have 1 bit of memory, in order to read it out and reset it, no matter how smart you are, you need a certain amount of heat entropy equal to kT log 2, where T is the environment of the bit reading object. In our case, our motors work at body temperature. Those can work at whatever temperature. These really small energy. And for those of you who follow the literature, this is an excerpt from Wikipedia. Landauer's limit, supported by Bennett and others, began to be attacked and say, well, this is kind of circular reasoning. We're not really sure this is a theorem, but it's an interesting proposition. And so that's where the Landauer limit stands, but it's changed. And the reason it's changed is because of all the advances in non-thermal equilibrium systems. All the stuff that was kind of on shaky ground is now rigorous. But the fundamental thing is the act of computation is inherently nonlinear. You're putting in power and you're doing computations. That's not thermal equilibrium. The cell is inherently non-thermal equilibrium. You're putting in chemical power and doing something. And so most of the systems, what we really care about is a non-thermal equilibrium. And so paying attention, especially to these steady state situations, you can get a lot. And through this deeper understanding of nonlinear dynamics, Landauer's thing is true. But it required that. Okay, so I don't know how long I've been talking, but I just want to say, you've just learned, if you improve measurements by 2 orders of magnitude, you get to learn a few new things. Our current work, we can improve things by another 2 orders of magnitude. We believe we can measure the displacement of this vesicle to a few angstroms in a live cell. And with a time resolution of at least 10 microseconds better, maybe 1 microsecond. So we're building up to do that. And at that point, you can see all the little micromotions that we think are there. We've invented, these are nanoparticles, so we invented ways to get nanoparticles into cells, thousands and thousands of them, into the cytosol of cells. The only way previously you get nanoparticles into cells is you drop them on the surface and the cells eat them, they endocytose them, but they end up in the stomachs of the lysozymes of cells. And the ability to get nanoparticles into cells opens up lots of new things, vast regions of biology. Just one trivial example, if we stick magnetic nanoparticles into immune cells and then stick them back into the human body, these are iron oxide particles. They can be imaged by MRI and other means of magnetic imaging, so you can look through the whole body. And so you can trace actually where these immune cells go. Can they find the cancer? Can they find the inflammation or something? And for those of you who know, there's a new therapy called CAR-T, T for killer T cell therapy, where they take the immune cells out of your body, they genetically modify them so they recognize your cancer, your blood cancer. They grow them up to huge quantities, they stick them back into the patient, and the patient nearly dies for a couple of weeks because of the immune reactions. But in the end, in blood cancer cases, in 2/3 of the cases, the cancer's gone and hasn't come back yet. Yet means only 3 years. This is pretty new stuff. But it can't find solid tumors. And there are other mysteries of this. So we think our nanoparticles will be good for all this stuff. Let me just close by thanking the people. Sam Peng was the postdoc. He and I worked on this for 6 years. The last 3 years, understanding the biology. The beginning, this day was taken by Sam, Yunsheng, Quan Lu, and Abe Barney came to light and helped in some of the discussions. So Sam is now beginning as assistant professor this fall at MIT. Yunsheng and Quan Lu are at Fudan University at Marty's. Bianxiao Sui helped Sam by teaching him how to take fetal neurons and put them into flow cells. She was in my group as a postdoc in 2006, where she was doing transport, the same transport using quantum dots, and we got frustrated because the quantum dots blink. So when we got the new particles, the first thing we wanted to do is do the experiment all over again. And then finally, Tom Sudolf supplied us with the human ion cells that have a predisposition to early Alzheimer's. The funding flowed in the usual way. No one wants to fund you, so you get an internal seed grant. After you get the seed grant, you can get an external seed grant. And after you get the external seed grant and you've done the work and you say, here's the preliminary data, i.e. the preprint, the NIH funds you. All right, so anyway, thank you for paying attention and happy birthday, Manny.
Thank you for that absolutely fascinating talk. That was really wonderful. Are there any questions? Maybe Lance?
Yeah, Steve, could you explain again how the forward biasing of the system works? Why does
Yeah,
it
great
tend to
question.
go one way and not the other?
Great question, great question. If you look at the structural data, when it releases, there's a linker arm that bends. And what it does is, if you just look at the structural data and it's attached to something else, this linker arm bends and there's a structural change and it kind of biases it. If it's here, it biases kind of over here. Now, by looking at the dimensions of the structures and the angles, which have been measured by other people, we calculated that this thing biases forward by 10.8 nanometers. Okay, now if it's doing a search about 10.8 nanometers, sometimes it'll land forward, sometimes it'll land back, but the mean is 10.8 nanometers. But when it finally makes a step, the other thing hasn't stepped. Therefore, it's 5.4 nanometers. What we measure is 5 nanometers. So it was purely of structures, published structures, and just taking the logical thing. And so that's what was so gratifying about this. It was all in the structures. No one bothered to stick it together and apply freshman physics. Sophomore physics.
Thank you very much for a great talk. So you mentioned a little bit about the technology needed in order to achieve that, and you say that you now are hoping to have a factor of 2 improvement. Would you?
No, 2 orders of
Two
magnitude.
orders of
We
magnitude.
don't do factors of 2
All right.
in my lab.
That's amazing. And so are you going to be able to do it with the same technology for quantum dots, or do you need to change the technology?
Yeah.
for that?
So the nanoparticles, so we're getting 5 million counts a second, 2 orders of magnitude, which you have to go up 4 orders of magnitude in counting rate. And we can't get that with our particles, but there is a way to do it. You can take a gold-coated shell, which is coated on polystyrene, so it's a thin layer of gold, and you can find its plasmonic resonance can be very high. And so it turns out that a 150 nanometer plasmonic shell, not a solid gold, but a shell, has a very good resonance. And we calculate based on intensity we're now using in our cells, so you're not going to kill the cell, the amount of scattered light should be about 5 microwatts. So we're no longer counting photons. Now, the noise floor of the balanced detectors we're having has a noise floor of 2 picowatts per root hertz. So if you want to detect at 1 megahertz, it's up by 3 orders of magnitude, so 2 nanowatts of noise with a bandwidth of 1 megahertz, which is about a half a microsecond. Okay, and that gives you ostensibly a signal noise about 1,000 to 1. Well, you only need 10 to 1. So we have 2 orders of magnitude to throw away before the system fails. And so we're building up to do this. And in this case, we'll have an automatic tracking system. So the tracker just calmly glides along with a low-pass filter. And then the high-pass filter is measured by 4 low-dose photodiodes. You split the beam up, and it jiggles back and forth. And so you see the jiggling, but you can track the thing continuously for the centimeter. And so that's the idea. It's just standard atomic physics that people know how to do. Biologists maybe not know how to do this, but we do. And so that's the idea. The beautiful thing about that is we do really want to see down to a couple angstroms, at least a nanometer, because there's lots of little motions that we're not capturing.
Okay. Oh, maybe just one. Yeah, one more.
One neuron, we look for 20 minutes, the bazillion steps in those 20 minutes. So the statistics are built up because we're tracking for a long time. That's the other thing very different from all other tracking experiments where the thing finally blinks or photobleaches, is that they concatenate all sorts of things. And what's different is we know it's one vesicle, and we know it's the vesicle because we watch it go 900 microns. And that's it. And so, and because these things don't photobleach, you can look forever. One of the primary new experiments we're going to do is tag with smaller particles, proteins, and watch what they do in cells. The APP precursor protein, which when miscut, it folds into A-beta oligomers that form A-beta plaques that are found in Alzheimer's disease. But once you form a plaque, the neurons are dead. We don't even know what APP precursor does in a cell. We don't know its biological function. So we're going back to grade 0, Tom Sudoff and I, he got a Nobel Prize for identifying the molecules that involve in vesicle fusion for neurotransmission. And the ground zero is stick a probe on this thing and watch it. What does it do? What other proteins does it interact with? So we can make a guess as to what its biological function is. But when it goes bad, end stage stuff is bad. Similarly with the tau protein. When it goes bad, so it could be a lot of things, a lot of theories, but, so. Depends on the patience of the graduate student or postdoc. But I would say 10 to 100 is probably what you need to get reasonable statistics. So, but the statistics of the one is a long-term statistic. So all the distributions are just that one, which never has been done before because you didn't have these probes.
Okay, we should continue the discussion in the coffee break after the next talk. So we should thank Professor Chu one more time. That was great. Absolutely. That was fantastic. So